understanding what's going on, formulating sensible scientific questions to ask about it, and then looking at some of the statistics that you can use to actually look for differential methylation. So if we can start by just looking at the data that you've got to work with. So what you had at the end of Felix's talk were two main kinds of data. So you have the individual sequence reads. So those would be the sequences that came off your sequencer mapped against the genome. So you know where in the genome those came from. And then within those reads, at the point where you passed over a cytosine, you would have individual methylation calls. So that would be data that said for a given cytosine in an individual read, whether that cytosine was actually methylated or unmethylated. Okay? So those are the two things that you've got to work with. And in terms of the files that come out of Bismarck, you have the BAM file, and the BAM file gives you the positions of the reads in your genome. And then you've actually got two different formats for the methylation calls. The one that we would normally use is this coverage file, and the coverage file summarizes all of the methylation calls that you get for each cytosine. And then you get some slightly more detailed breakdown in these text files that give you more details, but it's the same information. When we come to analyzing this, there are a couple of big decisions to make very early on in your analysis. Okay, so both of these go around how you're going to treat your data for the rest of the analysis that you do. One of the choices that you get to make is whether you're actually going to look at all of the methylation calls or not. And quite commonly, you won't actually look at all of the calls that are in your data set. What you will often do is that you will only look at methylation calls which were in CpG context. So that means you had a cytosine immediately followed by a guanine. The reason that you normally care mostly about these is that context of cytosines immediately followed by a guanine, are the only ones who can maintain their methylation after a round of cell division. So when you copy your DNA, the newly synthesized DNA is always unmethylated. And the way that the cell propagates methylation from cell cycle to cell cycle is by copying the methylated strand onto the unmethylated strand. And that only works when you've got something to copy it to. So if you have a cytosine in CG context, there's another cytosine opposite the G on the other strand, so that methylation can be copied and maintained. In other contexts, the methylation gets lost after cell division. So what generally happens is that in any context other than CG context, your entire genome remains completely unmethylated because the methylation just washes out with a cell cycle. If you're working in plants, then the same mechanism exists, but it can work where you have an intermediate base. So if you have a cytosine, something else, and then a guanine, so plants can do that, animals can't. And other states called CHH states are generally unmethylated. So most of the time, it makes sense to only work with CpG context cytosines. And that also has the advantage that it allows you to throw away very large portions of your data, because the CpG cytosines are a small subset of the total, and it gives you a much more manageable data size to work with. The other decision you get to make very early on is how you treat the methylation calls from the two different original DNA strands. Here you have a top strand and a bottom strand in your genome, and you don't necessarily have the same methylation pattern on the top strand as you have on the bottom strand. But again, we normally make the assumption that in CG context, the methylation is likely to be symmetric between the top and bottom strands. So again, you don't have to, but most of the time it makes sense to analyze only the CG data and to merge together the information on the top and bottom strands to give yourself a single, simpler data set to work with. Okay? So those two decisions are something that you would make up front and will have a big effect on how much data you've got to deal with and the complexity of the analysis that you are going to deal with. What you should do for any data set, but very much so for any sequence data set, is that as soon as you've got the data, before you try and do any analysis, you want to just find a way to visualize it, and look at it, and understand what the structure of the data is. So it's worth thinking about what you should expect to see from bisulfite sequencing. So this is a screenshot looking at the reads. So this is not the methylation calls, this is just the reads out of a whole genome bisulfite sequencing experiment. So in the figure that you can see, the bars across the top are just genes. So this is a big chunk of mouse chromosome 1. So you can see the genes there, and then the red and the blue dots underneath, each one of those is one read from your data set. And what we should expect to see in a bisulfite data set is that we cover the whole genome pretty equally, okay, all the way across, because we're not selecting, we're not biasing, and that we should see from a read perspective both the top and bottom strands equally, okay, because again, we're not selecting. So you should have a look and see if that's true for your data and make sure it's behaving the way you expect. You'll probably find some places where that's not true. So for example, this is a part of, again, mouse chromosome 17, and you will find that there will be places in your genome where strange things happen to your data. So like for this part here, there was a small region where we ended up with about 600 times more data than we should have had. And those are regions that we would generally try and identify and remove, or to at least remember where they are, because if we get something that turns out to be a hit from there, because they're behaving so weirdly, we would probably consider them to be unreliable and not somewhere where we'd actually want to take results from. So just looking at your data, understanding the structure of it, and if there are strange looking parts or parts with unusual coverage, then identifying those and just sort of keeping a note of those, so that if the hits that we get later come out from those, we can choose to maybe ignore those or investigate them more closely. You also might want to look at the coverage if you're ever comparing data from more than one bisulfite kit. So we said that the expectation is that we see all areas of the genome covered equally, but again, that's not true. Different kits will have different biases. So what I'm showing here is a data set which I've quantitated just with the number of reads that go across a region. So again, this is just part of a mouse chromosome. And you can see that the coverage isn't completely flat. The bars in here go up and down a bit, not hugely, but they do go up and down. But we sequenced this same sample with 2 different bisulfite kits, just from different manufacturers. And what you can see in this case is that the sequence biases that we get, so the places where we have high coverage in kit 1, are actually the places with the lowest coverage in kit 2. So if you're just using the same kit for everything, it's not so bad because the biases tend to be consistent. But if you're comparing between kits, then you want to really understand this, because those biases might lead you to make false predictions of differences that actually just arise from the selection biases from the kits. So all of this is really just coming about understanding and being comfortable with what the data looks like. Finally, both of those previous screenshots were from whole genome bisulfite, but sometimes you're not doing that, you're doing reduced representation sequencing, and for reduced representation you would expect your data to look very different. So this is a screenshot from some reduced representation data, and now you can see this is what you're getting when you're doing reduced representation. You don't now see complete coverage of the genome, you see large portions of the genome where you get no data at all, but the places where you do have data, you have quite a lot more. Okay, so this is the trade-off you make with reduced representation data. You don't see everything, but for the same amount of sequencing, you get much higher depth of coverage in the places in your genome where you really care about it, which is normally sort of CPG islands or high CG regions. Okay, so finding a way to visualize your data, look at it and be comfortable with the structure of it, and spotting if it's behaving the way that you expect is kind of the first quality step in understanding what's going on. After you've looked at your data, one of the first steps in actually analyzing it is going to be to quantitate your data. So normally the value that's used to quantitate a region of genome is the percentage methylation. So you have a lot of individual methylation calls but we normally collapse that down, so you end up with a single numerical value saying what percentage of the calls for a given region were methylated and unmethylated, and then comparing those numbers between samples. And calculating this percentage methylation value can be quite tricky. It would be really easy if all of your genome looked like this. So in this view, each dot on here is one methylation call, and the filled in dots, like the black dots, are methylated and the white dots are unmethylated. Okay, so standard representation of this data. And in this case, calculating the percentage of methylation would be easy. You just say, well, there's 15 calls in total, 10 of them were methylated, so therefore my methylation level for this region is 60%. Okay, that would be fine. However, the genome doesn't all look like this. You get cases like this. So here, you can see that there's an obvious difference in the depth of coverage across different parts of the region that we're quantitating. So if this was a reduced representation, we're specifically making parts of the genome better covered than others, but even in normal bisulfite, you get variations in the depth of coverage. If we calculate the percentage methylation by just putting all of these calls together and say, well, how many methylated and how many unmethylated do we see, then overall this region would come out as 46% methylated, despite the fact that most of the area of the region is completely methylated. So, alternate to just throwing everything together, you can calculate your percentage methylation per base. So you would say, well, this is 100% methylated, this is 100%, this is 100%, this is 100%, and that's 0%, and then averaging the per base methylation percentages to give you a value of 80% in this case, which would make more sense. So just to make you aware that depending on the choices you make about how you quantitate your data, you can get answers that sound really quite different just from the choice in how you quantitate. Okay? And this has nothing to do with the data, it's just the way it gets collapsed. Sorry, I can hear an awful lot of sound coming out of your microphone. Could you mute your thing? Okay, lovely. Thank you.
Sorry.
That's all right. So the reason that this matters is that when you're looking for differential methylation, that you can misinfer that differences in coverage could appear that they are differences in methylation. So in this example here, I've got two samples that I'm comparing, and I'm looking for differential methylation. And what you can see is that in the places where I measure the same base in both samples, they're both exactly the same. But there are some bases that are only measured in the top sample, and there are some bases that are only measured in the bottom sample. So if I calculate the methylation percentages, it would appear that the top sample is more methylated than the bottom one, even though at an individual comparison, there's no difference at all. So how do we cope with this? Well, there are a few different ways. The easiest way is to say that when we're comparing samples, we only take bases for quantitation which were observed in both of the samples. So now we'd only take the ones with the red bars on them. So we're comparing like with like, and now we would see effectively no difference between the two samples. Okay? This works really well if you only have a couple of samples, but if you have a large number of samples, it doesn't scale. Because different bases will be randomly dropped from different samples, and if you had like 20 samples, you would lose a very large portion of your genome from doing this. So in those cases, you either just have to live with the artificial differences that get inferred and work out when you get your hits whether that might be the cause, or you do something where you try and impute the missing values to give yourself more comparable values. And just to illustrate that this isn't just a theoretical concern, here's some real data. So this is a scatter plot of two samples where we're plotting the methylation level in one sample on the x-axis, the methylation level of the other sample on the y-axis, and each one of these dots in here is a region that we measured in the genome. And you can see that generally the two samples are pretty similar. The dots all sort of sit on the diagonal. I've picked out one particular point, so this is the most changing point in the data. So this has a methylation level change of what, about 40% between the two samples, so quite a big change. We'll come on to statistics later, but this sample also has a hugely significant p-value, 3.2 times 10 to the minus 7. But if you go and look at the data underneath that point, it looks like this. So the representation I'm showing here now is looking at methylation calls rather than reads. So the red dots on here are methylated calls, and the blue dots are unmethylated. And then we have the first sample and the second sample. And you can see that the quantitative values for this, the bottom sample is 94% methylated over this area. The top sample is 47% methylated. But you can also see that where you directly compare these samples, they look exactly the same. And that the reason for the difference is there's this whole area at the start of the region where we get a lot of unmethylated calls in the top sample, which just weren't measured in the bottom sample. So the details of how we quantitate and how we interpret the methylation calls that we've got can sometimes make it look like there's a difference when actually there isn't. So being careful about how you quantitate and being as conservative as possible at quantitating, we talked about a couple of different strategies there, can make a big difference into how you interpret this data. You also need to decide where you're going to quantitate. So ideally, in a methylation data set, you quantitate every individual cytosine and compare those, but practically speaking, that's just not possible. You don't generally have enough data on each individual cytosine to be able to make an accurate measure, and you would end up with huge numbers of measures with very poor accuracy. So in practice, we have to collapse a number of cytosines together to give a methylation value over a region of the genome. And we get to decide how that works. So if we have a whole genome bisulfite, usually we would want to start by doing some kind of unbiased analysis, so tiling windows over our whole genome and then measuring those in an unbiased way. And then if the unbiased analysis shows that maybe all gene bodies were actually the interesting parts, we can go back and target those. But it's always best to start without any preconceptions about where your changes are going to be. In some cases, you have to target. So, for example, if you're doing reduced representation, there's no point doing unbiased analyses because you're not seeing the whole genome. So then you might pick targeted regions such as CPG islands or gene bodies or promoters. Okay, so if you can, it's always best to start unbiased and maybe move on to targeted. But sometimes you have to start with the targeted because of the way that you constructed your experiment. The big problem that you have when you're trying to analyze methylation data is that we're collapsing our calls down to quantitative values, this percentage methylation values, but variations in coverage can mean that those values are much more or less reliable. So for example, if I have two regions of my genome here, region A and region B, and both of them are quantitated as 50% methylation, I might consider them to be reasonably equivalent. But if I look at the data that I have underneath them, region A might only have a single cytosine in it, okay? Which means that yes, it's 50% methylated, but off a very small level of observations, so therefore that measure is not very accurately quantitated because even a single change in that would have had a big effect. Region B, on the other hand, had lots of data in it. There's lots of cytosines, or it's a big region. And although, again, it says 50% methylation, that's actually quite an accurate and stable value. So this variation here can arise even if your region A and region B are the same size, because you get wild variations in CG density going across the genome. They are not evenly spread. They tend to occur at very high density in small parts of the genome and in very low density over most of the genome. And it would also turn around because you would have variations in coverage depth. So again, if you had some kind of enrichment and there was just more data over region A than region B. So what does this mean for us? Well, it means that when we're constructing our analysis, it's a good idea to try to make your data as evenly powered in each window that you analyze as possible. So what you want to try and do is to construct measurement windows that, as much as possible, have the same amount of data in each window to try and keep your noise and your statistical power consistent across each window that you're comparing, so that you're making a fair comparison. I've shown in this graph here, each dot on this is one gene from a genome, and because the genes are obviously quite different lengths, I've plotted out the length of the gene, or the log length of the gene, against the observed variance that we saw across a set of replicates. And what you can see is that there is a strong relationship between the two, that the smaller your gene is, the more noisy the measures that you make are, and the longer your gene is, the less noisy the measures you make are. So comparing values of different genes gets quite difficult because the noise profiles and the accuracy of measurements are so different. So it makes your life a lot easier and makes your data much more interpretable if you can avoid this bias by doing your analysis in a way that gives you the same amount of data in each window to start with, okay? Because then everything would sit at the same point in this part of the graph, and you would have much less problem with differences in variation. So when we're doing our analyses, normally the way that we would do an unbiased analysis is that instead of taking fixed size windows, we take windows of a fixed number of cytosines or a fixed number of CpGs. Okay? So that would mean that the size of the analysis window would change. If you were over a CpG island, it might only be a few hundred bases. And if you're in an intergenic region, it might be sort of 5 or 10 kilobases. But actually, you can see in the graph that I showed below what sort of range of difference you get. So over CpG islands for 50 CpG window lengths, you get an average length of about a kilobase. And when you're outside CpG islands, it only goes up to about 2 kilobases. So it's not such a wide difference, but doing that gives you much more consistent values and makes it a lot easier to interpret what you're seeing when you're visualizing your data. Okay, so that was sort of looking at how we can quantitate this data. Let's look at how we actually visualize it. So it's important that you visualize your quantitative values to understand what the sort of global pattern of methylation is in your sample, and to be able to compare between samples. In the length of time that we've got today, I'm not going to go through in any detail how we actually do the quantitations and visualizations, but I'll give you some links at the end to show you how to get to information where you can actually do this stuff for yourself. The software that we're showing, all of the screenshots that you'll see in this came from a piece of software called SeatMonk, which is developed by us. The URL for the project page is on the bottom there. And SeatMonk is a graphical program, a desktop program that you can load your coverage files in, see all these sorts of views, quantitate your data, and do the statistical comparison. So it sort of works end to end for doing this sort of analysis, and everything that you're going to see was constructed in that. We've got a lot of tutorials for actually how to sort of practically use SeatMonk for this. So if you actually want to get into this, then you can go and look through those and see how to sort of use the interface with the software to do it. But today I'm going to focus on sort of theoretically what sort of things to look at. So the thing that you should do then is, having quantitated your data, you want to view that alongside the raw data. Okay, so again, if you look at this view, the top part of each of the tracks here are the methylation calls. So remember, the red ones are the methylated ones, the blue ones are the unmethylated ones. And then the bottom bars are the methylation percentage values that we've quantitated over each region. So what you can hopefully see, or what you should be able to see, is that the two values make sense when looked at against each other. So here you can see the methylation is quite high, and you can see mostly we're getting red dots. But at the point where we get to a CpG island down here, we can see the methylation percentage measured drops and then rises over the CpG island. And we can also see the data under here. We can see a big load of unmethylated calls which appear and then drop away. Okay, so always, you should never see anything surprising out of the quantitation. You should see that the raw data and the quantitation match up with each other, and then by putting multiple samples up, you can start comparing them. And I can see here that actually the patterns between the two different conditions here look pretty similar over this region. Okay, so we can look at it in the genomic context. We can also start summarizing the methylation. So one thing that's very interesting to look at always is the distribution of methylation levels across your genome. People often talk and often cite values for the average methylation level for a genome. So they'll say what the mean methylation was for the genome as a whole. But those sorts of values are actually not a great summarization of methylation. So if you look at the distribution of methylation levels in a genomic sample, you find that it's not a single distribution around a mean. Methylation tends to be very bimodally distributed. So you have a large number of parts of the genome which are completely unmethylated, and another large portion which are completely methylated, and actually very little in between. So when you're comparing, when you're sort of summarizing your samples, if you say, well, the mean methylation level for this sample is probably about 60% or so, but 60% is a value that almost nothing in the genome has. And you've got a lot of stuff that's 80 or 90% and a lot of stuff that's 20% or below. One of the things that you want to look at very early on then is what this distribution looks like in your sample and whether it changes between samples. So for example, in this case, I'm comparing two samples here, and what I can see is that the shape of the distribution is about the same, but that the proportion of regions that fall into the high and low methylated blocks shifts. So in this bottom one, there's more data in the high region and less in the low region. So what we're looking at are a set of positions that do a state change from low to high. In other cases, you might see something more subtle. So this is another way of looking at the data. So this is just looking at the methylation percentage on the y-axis against the percentile through the data. So just ordering your data from low to high. And in some cases, you see a very sort of sigmoidal curve. That will be like this, where you get a lot of low stuff and a lot of high stuff and not much in between. But other samples show a much more gradual move between low and high. So it's a fundamental shift in the distribution. There's other ways to show this. We quite like this view, so doing sort of beam plots of the data. And here you can see, again, fundamental differences in the patterning of methylation. This is looking at methylation reprogramming, so we expect that it's going to be quite different. But looking at it in this global way, if there's a global change, you want to understand that before you try and get in and look at individual regions, because obviously if the sample as a whole is changing, that's the big biological picture, and actually it's not so much what's happening at an individual level. So always look at your overall distributions before you get into anything specific. You can also look at pairwise comparisons, and pairwise comparisons will give you a very good impression of what sort of questions you should be asking of your data and how they might relate. So in this case, we can obviously see that the two samples are very similar to each other. Overall, there's a strong correspondence between the two, and overall they have the same methylation level. Okay, and just doing a simple pairwise comparison will do that. The software, the SeqMonk software, also makes it possible to interact with these kinds of plots. So if this was actually in the program, I could just double click on one of these outlier points here to be taken to the chromosome view to be able to see the underlying data to see why that was similar or different. Okay, so again, this all comes under the exploration and understanding of your data. In some cases, we get more dramatic shifts. So for example, here we have a treatment which greatly affected the global level of methylation, and this sort of pairwise plot will very quickly show you that that's what the actual story is and show you the degree of difference between the samples. Okay, so what I'm trying to stress in all of this is that when you're approaching this sort of data, you need to understand it. So you need to be able to visualize it. You need to be able to look at both the raw data and make sensible decisions about how to quantitate that. And then from the quantitations, you can then start looking for global differences, or indeed sort of start looking at individual regions of the genome to try and understand that in more depth before you then get on to the second part that we're going to cover in the talk, which is to look at asking specific questions and assessing those statistically. Okay, so I'm going to stop briefly there to see if you have any questions at that point before we get on to the actual differential methylation statistics. So if there's any questions at that point, I'm quite happy to take those now.
Okay, I got questions. Whether I just wonder whether if we implement to visualize the methylation course as like you implemented in Sigmund,
Yep.
how we can implement actually for the whole genome is quite loading the data into the plotting the data, whole genome data is quite long and
Yeah, well, you're never going to plot the whole genome. I mean, there's just way too much data. But things that will turn up will either be completely universal. So if there's some kind of problem in the way that the data has been processed or that it's not behaving right, just sort of randomly scattered, looking around the genome at different parts, you'll probably see that very quickly. Okay, so to some extent, it doesn't necessarily matter. What we would always say for, again, any kind of data is that the places you should be looking are, firstly, just randomly in the genome, just to see what's there. Secondly, you should look at anywhere where you think you know what should be happening. So if you are looking at a methylation sample and you know that there is a particular region which is supposed to be differentially methylated given the samples that you look at, go and look at that, okay? Because that should either match with your expectations, or if it doesn't, then you've got something you need to understand. Ultimately, there's a limit to how much you can look at manually. So, or having done that, the last step at the end of the differential methylation would be that you get your set of hits, and before you do any activity on those hits, you should then take those and go back and look at those in the original data. Okay, so for example, in the differential hit that I showed you the example of, it would be very easy just to say, okay, well, that had a good p-value, it's got a big difference, therefore I'm going to put that in the paper, or I'm going to do more work to follow it up. But as soon as you look at the data underlying it, it's immediately obvious that actually it's not a real hit and it's a technical misprediction. So those then would be the places you have to look, and the initial view is just to spot large differences, which would mean it doesn't really matter where you go.
okay. Thank you. Okay, you can.
Okay.
Yeah.
Should we go on with the differential methylation stuff then? Okay, so differential methylation isn't the only thing you can do with methylation analysis. Sometimes you're just interested in a single sample, but a lot of the time what you're looking for are methylation changes in response to something that you've done. And in theory, again, it's a pretty simple question. So now it's the same sort of view as we had before, where we have individual samples with methylation calls which are either methylated or unmethylated. And now we have two samples where we're trying to compare the methylation calls between the two. And the question that we're asking is, are the calls that we get from the two samples consistent with them coming from an underlying methylation population which is exactly the same between the two? Okay? So in this case, we can see that there's more methylation in the top sample than the bottom one, but what the statistics are there to tell us is whether we could have seen something that looked this different just by random chance through sampling variation. Okay? So that's what we're trying to address in our statistics. There are two kind of main strategies that you can apply to do differential methylation statistics. So starting from your raw data, you can either go down the line of quantitating percentage methylation values and then doing continuous statistics on the quantitated values. That doesn't give you an awful lot of power in your data because you're reducing each measurement down to just a single number. And we've already talked about the fact that there are problems because the noise that's associated with those values is not constant across your data. But it does allow you to do a lot of normalization or correction that might be difficult to implement in other ways. The alternate way, which is the probably more common and certainly the more powerful approach, is that you use count-based statistics. So what you take for a region that you're comparing is you count how many methylated and unmethylated observations you have, and then you compare those ratios between different conditions that you want to analyze, and then use binomial-based statistics on that in order to treat that. The problem with that is if your values are not completely directly comparable, it's very difficult to introduce any kind of correction into this to account for global differences that might be there. There is a compound strategy, which I will also talk about, which is that you can do what's called reverse counting. So that means that you take your data, you put it into a percentage methylation value, you then correct that percentage methylation value, so you get an adjusted value, and then you infer what counts you would have got if you'd started with that percentage methylation value, and then you go down the route of doing the count-based statistics. Okay, so I'll talk about that in a bit, but actually that ends up being quite a useful strategy. So things that you need to think about when you approach differential methylation, you have to make sure that the question that you're asking is actually sensible. In some cases, you might need to apply corrections to your individual quantitations, that just taking the raw quantitation may not be the best course of action. With any statistical analysis, but really very much so in methylation analysis, you need to think about whether you've got enough data and enough statistical power to actually detect what it is that you want to see. And then finally, you want to try and see if there are any ways that you can make biological inferences out of the hits that you get. So let's start with the question that you want to ask. The question most people think of when they look at differential methylation is just to say, well, take the two sets of methylation values from two different conditions and directly compare them to say what's not the same between my two conditions. And if your overall relationship between your samples looks like this, that's a perfectly sensible question. You can see here that the overall methylation trend between the two samples is that they are exactly the same. They sit directly on the diagonal. And in this case, we could say, well, the points that are falling off the edge of this distribution might be the ones that are most regulated, and therefore we might be interested in those, and that would be what we would want to assess statistically. Sometimes that question doesn't make sense. So this is the same point that I showed before. We could ask the question of, well, what changes between these two samples? But looking at the global difference between them, we can see that pretty much everything changes between these samples. Okay? So that doesn't end up being a sensible question to ask. If we take an extreme example, if we take a point in here that's directly on the diagonal line, so it has exactly the same methylation value between the two samples, in a naive differential methylation analysis, that point would be considered completely uninteresting because it's exactly the same. But seeing what's going on in the rest of the sample, the points that don't change now become the most interesting ones of all, because why did they not change when everything else did? So we might want to restructure our question to say, well, what changed more or less than the sample as a whole changed, and then use that to identify interesting points. There's also a hybrid somewhere between the two. So this is another example of a methylation comparison. And now we can see in this case that there is a direct relationship between the two samples. So there is a fairly sort of straight line coming up here. It's quite noisy, but there's definitely a relationship there. But we can also see that that line doesn't line up with the diagonal on here. So the default position here is not that we see exactly the same level of methylation. There is a small global shift. And if we don't account for that shift and we ask what's changing, we're going to see tons of stuff that looks like it goes up in the data on the y-axis and hardly anything that goes down. So the question that might be more sensible here is, can we find the points which are changing unusually after correcting for this small global difference that otherwise would confound the analysis? Okay, so this all comes down again to doing the exploration properly and allowing yourself to formulate a sensible question. So what statistics can we apply to this? If we're dealing with count-based statistics, what we're essentially dealing with are contingency statistics. So in the simplest case, where we're dealing with just a pairwise comparison of unreplicated data, we construct a contingency table where for each of the samples for a given region, we count up how many methylated and unmethylated observations that we have between the two. We make a 2x2 contingency table, and then we can put those through standard contingency statistics. The likelihood of this coming up as different statistically will depend not only on the difference in the ratios between the two samples, but also in the number of the observations that we have. If we have lots of observations, count-based statistics will have more confidence than if we have low numbers of observations. And what that comes down to then is an issue of statistical power, okay, which relates to what we were talking to before. So again, it's unlikely that we're going to be able to analyze an individual cytosine. So we need to analyze in windows, and we have to make a trade-off when we're making our windows around statistical power. So the choices that we get, we have windows which can be either small or large. And what are the trade-offs between the two? So large windows have a lot of advantages. Okay? So we get lots of data per window, so we have high numbers of counts, which gives us a high statistical power because our power is going to be related to how much data we've got to work with. We also have to correct our statistical tests for the number of tests we've performed. So a p-value out of a statistical test is only valid if we only do one test. If we're testing 100,000 regions across our genome, we have to correct our p-values for the fact that we did 100,000 tests. So our p-values will be much higher than they're initially calculated at. The larger our windows, the smaller the number of tests that we have to perform, so therefore the lower the multiple testing correction burden is. So therefore it's easier to achieve low p-values. The only reason for not using large windows is if we make our window larger than the size of the differentially methylated region, because now we're bringing more data in, but we're also starting to average away the differential methylation effect that we're trying to detect. Okay, so as soon as our window size is bigger than the region which is changing, our effect size, then we're actually making things worse rather than better. Conversely, if we go to small windows, the thing that we gain is resolution. So we're much more likely to stay inside the window of change. Therefore, we're going to spot really quite specific biological effects. The downside for small windows is that we are going to have a lot of them, so therefore we have a high multiple testing correction burden. We're going to have less data inside each window because it's just a smaller area of the genome, and therefore it's going to be harder to achieve significance. So before you even get into doing the analysis, it's worth looking at some sort of pre-computed power analysis to see whether you're realistically likely to be able to see the sort of data that you want to see. The example I'm showing up here is a power analysis for a human genome, whole genome bisulfite, assuming that we want to see statistically significant points with p less than 0.05 and our power of detection, so we want about an 80% chance of spotting something if it actually changes. The table that you can see in here then shows us the size of region that we analyze. So this is numbers of cytosines. This will be analyzing each cytosine individually up to putting 500 adjacent cytosines together in an analysis window. And the y-axis on here shows the size of the methylation change. So if our reference methylation is 80%, would we detect a change to 79% or 75% or 70% or 60% and so on? Okay, so the increasing size. And the number that's in the table then is the fold coverage of the genome that you would need to have enough statistical power to do that analysis. So if somebody said, well, I want to analyze in a window of 10 cytosines and I want to detect anything up to a 10% methylation change, you can just look up and say, well, 10 cytosines and a 10% change, you're going to need to do 164-fold coverage of the genome in order to do that, at which point they will change their mind about wanting to do one of those things because no one's going to pay to do 164-fold coverage of the genome. Okay? So being realistic about what you can detect, it's very easy for a group leader to say, oh, well, I want to spot changes of 5%, but then when you're saying, well, you need to do 10x coverage of the genome to even detect a 5% change with 500 cytosines, they can become more reasonable about what's actually detectable. So if you had a high-seq lane, you might be up at sort of 3 to 5x coverage of the genome. So that would mean that you can detect a 20% methylation change in a 100 CpG window. So working out what's realistic for you to see is all part of doing the statistical analysis, and not being disappointed later on when you don't see what you think you should have been able to see. Okay. Statistics that we're going to use then for contingency table, we use standard contingency statistics. So for unreplicated data, you can use either chi-square stats or Fisher's exact statistics. It doesn't actually make an awful lot of difference which one of the contingency table statistics you use. The variation in the answers that you get from them tend to occur at relatively low observation levels. So for example, a chi-square and a Fisher's exact test, once you're up to about 20 observations per bin in your table, the two converge almost identically. And for a genome scale bisulfite experiment, anything that hasn't got that sort of number of observations in it, you're never going to see as significant anyway. So it doesn't make an awful lot of difference which one of those you actually apply. You can see from the example before, I've highlighted now the hits that come out of this contingency stats in blue, and you can see that it picks out the sort of things that you would want to expect, that it's going to be the sort of edges of your distribution and things that really change. The limitation with chi-square and Fisher's exact is that they are single observation statistics. So we would strongly recommend that people do biological replicates in their methylation data, and the extension of these statistics to replicated data is logistic regression. So logistic regression would allow you then to take count-based contingency data with replicates and take into account the variance that you see between sample groups and compare that with the variance within groups, and also taking the number of observations into account to identify things that change consistently. So in this example here, I've shown a heat map of hits that came out of a logistic regression analysis. So this is a 3 versus 3. So I've got 3 samples here in one condition and 3 samples in the other, and the colors simply represent whether the methylation level was higher in one sample or lower than it was in the group as a whole. So you can see there's a bunch of samples at the top which are all consistently higher in these oocyte samples than they were in the stem cell samples, and a larger group at the bottom which were consistently higher in the stem cells than they were in the oocytes. Okay, so that's what you're looking for out of these sort of stats. Not only that they're different in terms of their overall methylation level, but that they are reasonably consistently different within the replicates that you find in each condition. If you have a more unusual structure to your data, so this was the one where we saw a large global change, we can use binomial statistics. So what you essentially do in binomial statistics is that you change your default expectation from being that they should be the same to calculating a value by doing a regression through this distribution to say that's my default expectation, and you look for differences from that regressed line instead of looking for differences from the diagonal. Okay, so this would then allow you to find any point on this where the level of change was unusual, but it could definitely be different as long as it's not unusually different. And those sort of statistics would then allow you to pick out points like this, where you can see that there's a clear group which behave as the sample as a whole is behaving, and that the points that you pick out are those which retained more methylation than they should, or in some cases lost more methylation than you'd expect. Okay, so there are variations on the binomial statistics that apply if your question is different. There are some limitations with these sort of count-based statistics. So we saw when we were talking about the quantitation that one of the limitations is that you don't necessarily want to quantitate by just throwing all of the calls for a given region in together to calculate an overall percentage methylation value. But realistically, with count-based statistics, that's what you end up doing. So that's why reviewing your hits is always important afterwards, because something where you have very different coverage in different parts of your region can do that. There's also the issue of biasing by power. So this isn't a problem with the statistic itself. It tends to be a problem in the way that people biologically evaluate the hits that they get. We've seen several papers that do an unbiased analysis across the genome and then get very excited and say, well, we found that CPG islands were highly enriched. And then when you look at it, what you find is that the CpG islands were enriched not because they changed any more than the rest of the genome, but because they have more cytosines, and therefore you had more statistical power. So the same degree of change was more likely to be detected in the CpG island than it was in a region with low CG density. So just be careful about how you interpret if you don't have even statistical power. One final complication for the statistical analysis then is where you have a case like this. So here we have lots of different samples with methylation levels which are related, but we can see that there are global differences between them. If you've got differences in your methylation levels, it's often justifiable to normalize those away. So there's a number of ways in which you can do that. So in this case, I can do a fairly simple normalization by just picking a part of the distribution and then adjusting all the values so that they match at that point. So here I matched all of my data at the 85th percentile. So I've applied a constant scaling factor to all of my methylation values to make them more comparable. On a slightly more extreme level, you can also do things like quantile normalization, where you force all of the data onto the same distribution, which is kind of less satisfying academically, but produces a very clean looking result on this. But in each case, what you end up with then are normalized values which are not the values that you got from your original data. And this would then cause a problem when you actually want to do the statistics, because the counts won't reflect the quantitative values that you've normalized. So what you can do then is this idea of reverse counting. So in this case, I would have an example where I said that the actual observations that I had for a sample was that it was 40% methylated. So it was 20 methylation observations and 30 unmethylation observations. But after doing a correction and a normalization, that value then increased to 50%. Okay, in the same way as we did in the previous one. So what we could then do is to redivide those original counts based on the new methylation percentage, and therefore put counts of 25 and 25 into the count-based statistics in order to correct for that, but to keep the power of the count statistics. Okay, so the nice thing about that is that it adjusts for the corrections, but it retains the correct noise profile from the level of observation that it has. And this can give you quite different answers, but they can be quite biologically relevant. So here's an example that we had earlier in the year. So this is a comparison of two samples where it's not a big difference between these samples, but you can kind of see that this upper part of the distribution here is slightly skewed. So it doesn't sit quite on the diagonal, it skews down a bit. We actually know why this is in this case. It's because the sample here had been grown in culture for a few cell divisions, and when you culture things and cells grow quickly, the maintenance methylation doesn't quite keep up, so the methylation level can drop slightly, but for entirely uninteresting reasons. If you just do a standard analysis on this data, these are the points that come out as differentially methylated. Okay, it's the points at the bottom of this, and it's purely related to their original methylation level. It doesn't reflect the specific changes, it just reflects the global change in the sample. If I take the same sample and then I normalize the methylation levels, I can see that I can straighten this out. So if you can see it moving from there to there. And in the normalized data, if I then reanalyze that, I get a very different set of points. Okay, so there's a degree of sort of scientific interpretation in this, but the point is that by either applying that correction or not, you'll get a very different set of hits and you'll get a very different biological conclusion. So thinking about exactly what any differences you see are and making sure you're formulating your question correctly is going to be the key to actually understanding this. Okay, the last little bit then is just thinking about the biology of the hits that you get back. Statistics doesn't really care about the size of your difference. All it's doing is to say, is there enough statistical power to say that the difference looks like it's not technically consistent between the two. So whenever you get a hit back, think about whether you can actually posit a real biologically relevant mechanism for why, for example, a 5% methylation change might actually do something relevant in your sample. So what actually makes biological sense? If you have a set of hits You also then want to try and understand what might connect those hits. So some simple things that you can look for if you've done an unbiased analysis is, for example, do the hits relate to particular features? So do they relate to CPG islands? Do they relate to genes? Do they relate to promoters? And how do they cluster around features over your whole genome? And that might point you towards either a better way of analyzing the data or a biological meaning to the hits that you've got. You also might want to see if you're seeing sets of hits that fall adjacent to each other, because that again might give you a clue that instead of having lots of independent hits, you might be looking at larger hit regions. So just to illustrate that with a couple of examples, this was a region that we analysed last year. And when you look at it overall, they look pretty similar. So the methylation levels between these two samples, the profile and the level of methylation actually looks quite similar, but the statistics were actually picking out several points within here as being different, which were not very exciting large differences. But if I view my data again, not by looking at the absolute levels, but by plotting the difference between the two samples, I get this. So now, all of a sudden, my biology becomes a lot more convincing. So from a set of unaligned, not very convincing hits, suddenly I can now see that I get a set of hits which line up exactly with the gene body of this gene. And although individually they're not very big, the fact that they're connected and we can draw an inference between them makes them more biologically convincing. Finally, another way of looking at the same sort of thing is to do sort of aggregation of hits against features. So all this is to say, for the hits that I got, where did they fall relative to a gene? So this central part here is the area of the gene over a gene body, so over all gene bodies in the genome. This is then 5 kb upstream of the gene, 5 kb downstream of the gene, and I can see that my hits are very preferentially turning up inside genes, and maybe even more preferentially in the promoter of the gene. So at the start of the gene, I see a big spike in hits, but globally they're very much over genes. So again, showing me that I may not just be finding noise, but I might actually be picking out something with biological relevance. Okay. So I'm going to finish by leaving you a couple of links to packages that you might find useful. So like I said, the stuff that I've shown you has all been done in the free graphical SeqMonk package, which does all of the sort of visualization and statistics that we've done. It's an interactive desktop package, which is great for really getting into know and understand your data, and it's what we encourage people here to use. If you want to do a larger scale analysis and you need an automated way of doing it, a lot of the same sort of methods that we have in here are implemented in the methyl kit package, which is an R package, which has a number of different things that you can do in there, but the same sort of statistics are there. You'll need to construct your own visualization on top of that, or to take hits out and sort of bring those back into more of an interactive package to view it. But both of those are sort of useful tools to look at. If you want more practical information on how to do this, this link at the bottom is to our training part of our website, where we have all of the material for our bisulfite analysis course. So some of that will be the material that Felix covered last week and I've talked about today, but there's much more detail in there, and there are some practical parts of it where we go through how to run various bits of software. There's also example data sets on there and practical instructions so you can actually try and run this yourself and do it. If you have any questions, I'll take some questions now if anyone's got anything, but if you have other questions afterwards, my email address is also linked off that site, so I'm more than happy to have people email either myself or Felix. We're very happy to take questions after if you actually start getting into this data yourself. Okay, I'll stop there and take any questions.
Okay, if we use the different package analysis and then have different methylation region.
Sorry, so that I missed that.
Okay, if we use the different package, different software for the differential analysis, and at the end of the analysis, we can get the different result, different set of the signature region of the methylation from different.
Yeah, so you'll get different hits depending on a lot of the choices that you make when you put through. What you should see, though, is that although individual regions might not be in exactly the same place, or individual hits might fall in and out, the same biological theme should come out. So if your hits say that it's a certain class of genes that's changing or subset, that should stay robust through the choices you make. And if it doesn't, then I'd be concerned about how valid the conclusion I was drawing from that should be. So with any analysis, the exact choice of the statistics or the way that you quantitate it or the cutoffs that you use will always change the set of hits that you get somewhat, but that shouldn't change the conclusion that you draw at the end of it. And if it does, maybe it wasn't a very strong conclusion in the first place.
Yeah, I see. Thank you for the good suggestions. Yeah.
Okay.
Okay, let's see whether we have got the question from the other audience. We haven't got the questions yet. So
Okay. Where would they show up? Do they just come in the chat?
in the list of attendees, we can see also they can send a chat.
Okay.
So okay. My question is about the if we do differential analysis between the samples, two different conditions. Actually, we just compare the fixed length of CPG density, right?
So we would normally recommend, if it's an unbiased analysis, analyzing fixed data windows. So you've analyzed windows of fixed numbers of CPGs.
Fixed number.
Because then you get the same statistical power in each window and you get roughly equivalent noise profiles. Fixed length windows will give you some that have hardly any data in and some that have lots of data. So fixed data is always better than fixed length for this analysis.
Fixed length of CPG density.
Yes, fixed numbers of CPGs.
Yeah. Good. Okay, maybe we can end the session today. Just
Okay.
at this point. Okay, thank you very much for giving us the webinar.
No problem.
Okay, see you next time.
Okay, thanks a lot then.
Bye.
Bye.
Okay.